Hello students, welcome to Digital Learning DCE. Myself Dr. Manjula. In this session, let us study about tools involved in genetic engineering with special emphasis on restriction endonucleases and vectors. Gene cloning uses some biological products and agents for achieving its objectives. These are generally called as genetic engineering tools. Genetic engineering uses four important tools which are as follows. First one, restriction enzymes, which are commonly called as molecular scissors, cut DNA at specific locations. Second one, DNA ligase, which are popularly known as molecular glue, which create covalent bonds between nucleotide chains. Third important tool is vector, which are popularly known as molecular vehicles. They carry the desired gene from the choice of organism into the host. And fourth important tool is a suitable organism that serves as a host for propagation of the recombinant DNA. In addition to above mentioned four basic tools, several enzymes and DNA sequences are also used in genetic engineering as tools. Reverse transcriptase are a group of enzymes which synthesize DNA from mRNA template. Alkaline phosphatases involved in removal of phosphate groups at the 5' end of the DNA. T4 polynucleotide kinase. They transfer phosphoryl group from ATP to dephosphorylated ends of DNA or RNA. E. coli exonuclease 3 are the enzymes which involved in removal of nucleotides from the DNA strand. The clean-out fragment of E. coli DNA polymerase 1 involved in synthesis of DNA on a DNA template. Likewise, terminal deoxynucleotidyl transferase, linker and adapter oligonucleotide sequences are also used as additional tools in genetic engineering. Coming to restriction endonucleases, they are the important tools used in genetic engineering. They are the DNA cutting enzymes essential for modification of DNA. The generally used restriction endonucleases in genetic engineering includes EcoR1, End3 and TAC1. Since they make a cut within the molecule, they are also called as restriction endonucleases. These enzymes recognizes a particular target sequence and double-stranded DNA and next the polynucleotide strand within the sequence to give discrete fragments of desired length. They cut double-stranded DNA and make two incisions, hence the name molecular scissors. They are classified into three types like type 1, type 2 and type 3 endonucleases. In this slide, we can observe an example of restriction endonuclease, EcoR1, which is common bound to the double-stranded DNA molecule and recognizes the sites on the DNA segment where to make a cut and finally make two blunt sticky ends. Coming to the types of restriction enzymes. Type 1 restriction enzymes recognizes long specific sequences but cleave the DNA at random points more than 1000 base pair away from the recognition sequence. Here, both methylation and cleavage are performed by the same enzyme. Type 1 restriction enzymes includes EcoK1 and EcoB1. Type 2 restriction enzymes, here restriction and modifications are mediated by separate subunits of enzymes. Thus, it is possible to cleave the DNA in the absence of modification. It recognizes specific sequences of 4 to 7 bases long and cuts a specific place, hence most useful in gene cloning. EcoR1 and End2 are the examples of type 2 restriction enzymes. Coming to the third type, that is type 3 restriction enzymes, here methylation and cleavage are performed by separate subunits, which cleaves the DNA at random points about 24 to 26 base pair away from the recognition sequence. Example for type 3 restriction enzymes include EcoP11 and EcoP15. Coming to vectors as important tools in genetic engineering. A vector is a DNA molecule which is capable of autonomous replication and carry a foreign DNA fragment when it is inserted into it. The most popularly used vectors in genetic engineering includes PBR322 and PUC18. Any plasmid to be used as a vector should possess some important properties which are as follows. A vector should be a small, easy to isolate and purify, able to replicate autonomously. A vector should be easily introduced into the host cell. A vector must have one or more origin of replication and also more unique restriction sites. A vector should have a suitable marker gene which allows easy screening of the transformed cells. Finally, a good vector should not be toxic to the host cell. Coming to the types of vector, based on the nature and source, vectors are of different types like plasmid vectors, bacteriophage vectors, cosmids and phasmids. Let us study one by one in detail. Plasmid vectors. These are small extra chromosomal self-replicating double-stranded closed and circular DNA molecules present inside the bacteria. The most extensively used in genetic engineering when the small fragments of DNA have to be cloned. The most popularly used plasmid vector is PBR322. In this diagram, we can observe the PBR322 plasmid vector with origin of replication as well as two important resistant gene markers like ampicillin resistant and tetracycline gene resistant markers. Coming to the second type of vector, that is bacteriophage vector. It is a linear DNA molecule with a single break which will generate two fragments where foreign DNA can be inserted to generate chimeric phage particles. They can be accommodate large pieces of inserted DNA. Example for bacteriophage vector includes lambda bacteriophage and M13. In this diagram, we can see the M13 bacteriophage vector with origin of replication and promoter and terminator regions. Coming to the third type of vector, that is cosmids. Cosmids are hybrid vectors that contain cos site of lambda phage and a plasmid that can replicate like a plasmid. It is used to clone large piece of foreign DNA molecule of about 40 kilobase pair. Cosmids are generally used in production of DNA libraries of eukaryotes. In this diagram, we can see a cosmid vector which is formed by the combination of cos site as well as plasmid. This is a cos site and this is ampicillin resistant gene with origin of replication. Then coming to the fourth type of vector, that is plasmid, which is prepared artificially by combining features of phages with that of a plasmid. It is a plasmid that contain F1 origin of replication. The example for plasmid includes PYWO1, where we can observe the F1 origin of replication of the F1 phage. To sum up the session, gene cloning uses certain biological products and agents which are generally known as genetic engineering tools. Two important such tools used in genetic engineering are restriction endonucleases and vectors. Restriction endonucleases are DNA cutting enzymes found in some bacteria which recognizes and cuts DNA only at particular sequence of nucleotides. The most popularly used restriction endonucleases includes EcoR1 and N3. Restriction endonucleases are of three types like type 1, type 2 and type 3. Coming to the second important tool used in genetic engineering, that is vector, which is a DNA molecule which can carry a foreign DNA fragment when it is inserted into it. Example of vector includes PBR322 and PUC18. Vectors may be of four different types like plasmid, bacterial. For further details about the current topic, you can refer the following references. With this, I am going to conclude the session. Thank you.